So a very warm welcome to you for joining this UK Biobank webinar, which aims to highlight UK Biobank's cohort-wide NMR metabolite data, which was released at the end of last year. I'm Martin Rutter, I'm Deputy Chief Scientist at UK Biobank. We're here to promote the use of these data and highlight opportunities for research using these data. We'll also provide some guidance on how these data can be used effectively. As many of the people on this webinar will be aware, metabolic profiling can provide objective biological readout of combined effects of genetics, lifestyle, and environment, and it can be used to improve disease prediction, and it can also provide mechanistic insights into that will inform drug development, disease prevention and management, and also population health strategies. We have 4 fabulous speakers today, and I'll introduce the first 2 right now so we can save a little bit of time during the meeting. First up is going to be Luke Jostins-Dean, who's Principal Scientist at Nightingale Health and Group Leader at the University of Oxford. He works on building disease models for multi-omic data, deploying them to help clinicians and individuals manage patient health and he's going to tell us how these data were generated. Following on from Luke, we've got Lora Boteva, who's Senior Data Analyst here at UK Biobank, and she's going to tell us what data is available on our research analysis platform, how these data can be accessed, and the tools that are available for analysis. So without any further ado, I will bring Luke forward to give us his presentation.
Hi, thank you for the introduction, and thanks everyone for coming. So I'm going to be talking about how this data was generated and sort of what we're kind of doing with it. I'll give you have a quick section on what the data is, what biomarkers we measure, and why they're kind of interesting in various ways. I'll talk a little bit about the kind of resources that us and our collaborators have created for data users to sort of make the most of our data, both whether you are or are not an approved by UK Biobank user. And then we'll talk a little bit about the ways in which we're using this data sort of in-house in our own analyses to kind of learn things about biology and improve patient health. So our technology is an NMR metabolomics technology. It's nuclear magnetic resonance. It's a 1-proton NMR. We do a couple of different frequency peaks, and we have a number of peaks that kind of come out of that. And from those you can quantify around 250 biomarker or biomarker variables. We mostly quantify in absolute units. So we give you, instead of having a relative unit like you might get from some RNA or protein assays, this tells you exactly what the concentration is in millimolar per liter or micrograms per liter and things like that in sort of standard units. I'll talk a little bit about the different things that we measure, sort of a bit of an overview. A lot of what we measure is kind of really focused on lipoproteins and the sort of surrounding lipids. So imagine that a lipoprotein particle is made up of, has a large number of different lipids in it, and we measure many of those types. We measure total cholesterol. Sorry, I realize I need to. Apologies, I need to make this a lot bigger. Sorry, my screen is very small. So we measure various measures of total lipids, the kind of things you might be familiar with from your from clinical readouts from the doctor. Your cholesterol, amount of cholesterol, the amount of triglycerides in your lipid particles, the total lipids, some slightly more exotic lipid measures, cholines, phosphoglycones, things like that people are more interested in. And we also measure particularly detailed readout of the actual fatty acid composition. So the actual fatty acids that make up these lipids, which is the main component of these lipids. Those are your things like your omega-3s, your omega-6s, your degrees of unsaturation, your polyunsaturated, your monounsaturated fats, which are kind of important for health. We also measure apolipoproteins, the proteins that are embedded in this and help structure the lipid molecule. beyond just measuring just kind of total lipids, we do quite a detailed readout of the sort of lipoprotein classes. So lipoproteins can be of various different sizes. You've probably heard of HDL versus LDL. Some of the particles are very large, some of the particles are very small. We give actually quite a detailed breakdown sort of by, you know, the diameter of these molecules, the very large, the very small. Those are sort of biologically interesting, mostly because, or interesting medically, because some of these particles can enter the arterial intima and actually sort of contribute towards forming plaques and atherosclerosis. Some of them are too large to do so, and so they sort of have biological implications there. And we also then measure the sort of combination. We measure the lipid composition of each particle in each class size, so you can really dig down into what is the specific contribution of HDL, what is the contribution of very large LDL particles or the small LDL particles and things like that. I won't go into a huge amount of detail, but we do measure lots of other metabolites outside of the lipoproteins, various ketone bodies like your acetates, your acetones. We have quite a detailed measure of amino acids, particularly around the branched chain amino acids, but we sort of measure quite a lot of different amino acids. We measure glycolysis products, glucose, lactate, pyruvate. We measure albumin and creatinine, obviously important for sort of liver and obviously important for kidney function. And we also have this inflammatory measure, this glycoprotein acetylation, which is actually a kind of metabolic readout of chronic inflammation, and is quite a good marker for measuring, for example, future onset of inflammatory diseases. Okay. So we started measuring these in the lab around 2019. We started getting samples. Back in 2021, we released the first phase of 120,000 data from 120,000 samples to the UK Biobank on the showcase. In 2023, we released the phase 2 of the data, which is about half of the entire cohort that many of you may have analysed. And then just a few months ago, we released the full 500,000 samples. This is everyone in phase 3. There's also a lot of celebrations there. Some of you actually, I think, might have come to our celebration event in London to celebrate. This consisted both of the sort of main phase, the kind of baseline samples, about 490,000 baseline samples, which is essentially every sample that we could kind of get our hands on from UK Biobank. We also measured 20,000 repeat samples. So these are individuals that had a second measure taken around 5 years after the first one was done. We also measured about 10,000 duplicates. These were blind duplicates sent to us by UK Biobank for assessing quality. We didn't know what they were duplicates of, so we couldn't cheat. And then when we finished generating the data, UK Biobank mapped them back, and we could look at the actual quality of the results in terms of consistency. And we were quite happy with the quality of our data. This plot shows the sort of the correlation between two replicates of the apolipoprotein ApoB, and you can see a very good correlation. The dots in black are of the same, measured on the same spectrometer. The ones in red are on different spectrometers. They both showed a very good correlation between the blind duplicates. We did in the early stages have some markers that didn't look quite as good. So alanine was an example there, where in the first sort of batches we saw a pretty good consistency on the same spectrometer, but we were seeing noise when going from spectrometer to spectrometer that was causing increased error in that. We've since corrected that effect now, and now there is a spectrometer corrected alanine, which is available in the UK Biobank showcase as well. So that's now relatively accurate. That's not pretty accurate as well. So while the data has just been made available to everyone else, there have been a set of collaborators that have been accessing this data for a while. We've been publishing papers as we go along. We had a paper in 2023. That's probably the best paper for you to read if you really want to understand the sort of the methods that we generated the data, sort of the way that we quality control it, the way we sort of did basic kind of analyses of it, and that was on the phase 1. And then we had a paper last year on the full phase 3, and actually combining it with some other data sets to do mostly around risk prediction and comparing genetic and metabolic risk prediction. We also had a couple of sets of collaborators. Collaborators from Charity and QMUL published a nice paper last year from the early access to the data, mapping the effect of both rare and common variants on metabolites. And there's a nice big meta-analysis from the University of Tartu as well, which meta-analyzed our data, did genome-wide association studies of all our metabolites, and meta-analyzed them with the Estonian Biobank, and did a very large meta-analysis and discovered many thousands of associations. And all those summary statistics, as I'll say, are available that you can look up now and sort of look up the genetics yourself. So what data are we kind of putting out? There's the actual individual level data that's being released by UK Biobank via the RAP, which Laura's going to talk about in a moment. We have GWAS summary statistics from the collaboration with the University of Tartu that I just mentioned. So that includes meta-analysis of the UK Biobank between UK Biobank and Estonian Biobank, and also includes UK Biobank ancestry-specific analyses. So, 4,500 African ancestry individuals, GWAS for every metabolite, 8,500 for South Asian, some pretty large and well-powered non-European genome-wide association studies here as well. We've also made a website, so if you don't have access to the data, you can look up the associations yourself for individual biomarkers and diseases. So this allows you to look up any disease that we've studied for its association, metabolic association with prevalent disease, i.e. when you currently have it, incident disease, i.e. will you get it in the future, and mortality disease endpoints, i.e. will you die of them. That's available from that URL. You can download the raw data, and you can also make plots like these that I'm showing here, where you look up a particular ICD-10 code, and it gives you a dot for every metabolite with the association, and it fills in significance, and then you can highlight individual metabolites to see the association statistics. So type 2 diabetes is the top one. ICD-10 code E11, and you can see it's associated with almost everything with a very broad metabolic profile. Chronic kidney disease also has strong associations, but you can see down the left, you can see this one standout association that's strong with everyone else, which is the association with creatinine, which is a known, very well established biomarker of kidney failure. And finally, we also have some code and some sort of a worksheet that we've developed to help people learn how to analyze this data. It also includes some artificial simulated data that matches the sort of size and scale of the UK Biobank, but is all artificial data, so you can download it and kind of learn to use it yourself. It has code on how to do QC, how to do analysis. It's available on this Kaggle link here, and do take a look at it, sort of give you a nice introduction to all the data and how we think you should analyze it. Great. I'm going to quickly talk about a couple of the ways we've been using this data then, because I think we've got some illustrations of what makes it powerful. The first one is that we've been using this increasingly in the clinic and in practice to help guide disease risk assessments. So as well as having a look at all these individual associations on the left, the individual biomarkers, we can also construct multi-biomarker scores that take into account a weighting combination of all the different biomarkers. We take the UK Biobank, we split it into half, train models on half of them, test them on the other half of the UK Biobank. And what this produces is a statistical model that lets you go to every individual and produce a score that says this is the chance you're getting disease in the next 10 years, for example. And we can rank everyone to say you're at high risk, you're at low risk. And we can also we can go into UK Biobank and validate these scores and show this is I think this example is type 2 diabetes, that people at high risk develop disease at a much higher rate in the follow-up electronic healthcare follow-up record. And then we can divide people up, anyone up in the UK Biobank or anyone who walked in the door, into whether they're in a sort of high risk group, an intermediate risk group, or a low risk group. There's a paper on this process that you can read, and that we've put out that sort of has all that and has all the models in it and all the weights and everything if you want to use them yourself. They work quite well, and we've been using them particularly in cardiovascular disease, in type 2 diabetes, and chronic kidney disease, and also in various liver diseases. And you can, if you take the high-risk population as the top 10%, they all have increased risks of twofold, sometimes threefold, fourfold, even tenfold for some of these diseases. And these are all actually much better than the prediction you get from an equivalently sized set for genetics, which I guess makes sense because genetics captures only your inborn risk, whereas the metabolic score captures some of your inborn risk, but also environmental effects, your weight, your lifestyle. It's a measure of environmental risk as well as genetic risk. So we've been collaborating with a company in Finland. So Nightingale Health is a Finnish biotech company, and in Finland, a lot of risk assessment for common diseases is actually done through employers, through occupational health. That's sort of the role that many occupational health programs have in Finland. And so now we've been working with this company that provides one of these major sources of occupational health, and around 30% of the Finnish workforce is now receiving NMR-based assessments of risk. That's 200,000 people so far have received this results. And this company, they have occupational health nurses and doctors and a whole system of referral pathways to sort of manage people's risk. And we provide them with these reports where we, for example, for cardiovascular disease, we'll say whether you're at an elevated risk, low risk, high risk, and we'll also give measures that say how you are compared to your age. So if you're 30 and you're at relatively low risk of cardiovascular disease because you're young, we can still say you're at higher risk than your peers, and that might become a problem in the future. We've been assessing this kind of use in healthcare and collaborating with these kind of doctors and nurses to sort of see how well this is doing. We have a regular a survey that we do of healthcare professionals that are using our test and using these risk scores. We ask them various questions about how it's going. We've asked them, for example, whether these scores have been helpful in supporting lifestyle discussions with their patients, of which the vast majority agree quite strongly, and very few people disagree. So people clearly feel it's being useful, particularly in guiding lifestyle discussions. We also ask whether it's easy to present the data to the patients. You might notice here that there's a difference between nurses and doctors, where a large number of nurses have found it, a small minority, but still kind of in the, more than 15% of nurses find this difficult to communicate. So we've been working increasingly on making a new version of this, where we think not just about making the predictions, but how do we communicate it really effectively, and how do we make sure it can be fitted into people's procedures. I will skip through this bit quick fast because we're sort of running a bit low on time. It makes sense that, so we've measured metabolics now in 500,000 people. There's also detailed proteomic data in 50,000 people, and we've been looking increasingly at how to put those things together. We as a company generate metabolomic data, but we also generate for our customers and for ourselves proteomics data and various proteomic data sets. So we're interested in how you can analyze these two things together. In the UK Biobank, there's about 50 proteins that also metabolize the metabolites we measure. So you can do kind of direct measures of metabolism. But also we've been using the metabolic data to interpret the protein associations with disease. So this is sort of an example of that analysis. So on the x-axis is the p-value of association between various protein levels and the risk of future type 2 diabetes. And on the y-axis is the same analysis, but conditional on the full set of metabolites. So this is saying, what is the effect of proteins after you condition on the risk due to metabolites? And you can sort of see various different groups. You can see down the bottom proteins where the association goes away. These are explained by metabolic associations. ApoD is an example of that, where ApoD is a component protein of HDL. It is associated with type 2 diabetes, but that's entirely explained by its presence in lipoproteins. And if you adjust for the metabolite content, then there's no association with that. And on the other side, you have things where the protein associations are independent of the metabolic factors. These are proteins that aren't acting through measurable metabolic risk, and instead are entirely independent. And many of those are inflammatory, various cell surface cell integration things, various cytokine receptors, which are kind of interesting. And then finally, those are that are kind of intermediate, where they're associated with both metabolomics and proteomics, and both things give independent measures of risk. So we're measuring the same risk factors with a slightly different way of looking at it. All right. Just want to say thank you to everyone involved in this project, everyone at Nightingale who's delivered this, everyone at UK Biobank has helped, all our collaborators who have been doing analysis with us along the way, and then everyone in the research community and everyone sort of been involved in the UK Biobank. There's more than 300 papers have been written, R packages. People are attending webinars like this. So thank you a lot for attending now and for using our data, and I really hope that you make good use of the data in the future. Great. And now I'm going to hand over to Lora to talk a little bit about how you actually get the data access and what you can do with it on the UK Biobank side.
Hello, everyone, and thank you for tuning in. So I'm one of the senior data analysts here at UK Biobank, and I will briefly go over what data is available and how you can access it. So as of last November, we have NMR metabolomics data for the full cohort, so nearly all 500,000 participants taking part in the UK Biobank study, and that comprises raw data on 249 metabolic variables, including 168 absolute levels and 81 ratio measures. We also, as a part of this data set, have included QC flags and processing metrics, so things like time of processing or the spectrometer that was used to process that particular samples, and we have included those as for transparency purposes and also to enable researchers to filter and correct the data as they see fit for their specific scientific question. All of this is available under categories 220, 221, and 222 on Showcase. And if you do visit Showcase, you will also find that we have a number of companion resources covering sample processing, the ratio calculations, batch variation, and also details on how we have monitored reproducibility during the course of this study. There's a lot of very, very helpful resources, so I would really encourage anyone considering using this data and analyzing these data to visit Showcase and have a read through the resources. So data is available for nearly all of the cohort, about 98%, but also excitingly, we have longitudinal measures for about 17,500 participants. So that includes paired sample for the original baseline visit conducted between 2006 and 2010, when participants were aged 40 and up, and then an additional sample for the repeat assessment visit in 2012 and 2013. On average, I think there is about 4.5 years between the two collection samples. So obviously that enables a lot of really interesting longitudinal studies. So one of the most valuable aspects of the UK Biobank resource is, of course, that we have data across multiple modalities. And I have some data here on the coverage between different data modalities we have in the resource and the NMR study. So about half of the participants within the NMR study have GP record data available, but we are hoping to extend that very, very soon. About 20% of the participants in the study have imaging data available, so about just over 100,000, and that includes MRI images of the brain, heart, and abdomen, as well as things like DEXA scans for bone density and things like that. And that breaks down to about 80,000 participants who have just one imaging visit, but also an additional over 20,000 participants that actually have a repeat imaging visit, so 2 imaging visits as well, enabling again longitudinal analysis. So Luke briefly mentioned the O-link coverage as well. So 10% of the participants included in the NMR study also have data for O-link. So for those of you that are not familiar, this is a targeted proteomics affinity-based assay that measures about 3,000 proteins found in serum. And again, there is a possibility this might be extended in the future, both in terms of cohort coverage and the number of proteins assayed. And of course, we also have our whole genome sequencing dataset for the full cohorts, or nearly full coverage of the metabolomics dataset. And that spans everything from, the data available for that spans everything from raw sequencing files, so things like crumbs, to sort of more variation based file formats like VCFs for SNPs and copy number variants. So how can you access all that data? Of course, it's great to have a data set like this, but it also presents significant challenges. So the whole data set is about 25 petabytes in size, which is obviously quite a challenge for any individual research group to store, keep up to date, keep compliant in terms of removing withdrawals in a timely manner. So essentially, to try and balance usability, accessibility, and safety for our data, all of our data is now available just exclusively through our cloud-based research analysis platform, or RAP for short. So this ensures that data is stored in a safe and cost-effective manner. There is no direct cost to individual researchers for data storage. It also means that as long as you have a RAP account and a RAP project, you can get access to the latest data as soon as it's released and becomes available, and we look after removing withdrawn participants and things like that. There is a range of tools for every level of computational expertise, and because it's a cloud-based platform, it means that compute is scalable, and you can choose the right level of compute for your analysis, and you are also in control of the cost. In terms of getting access, you would need to submit an application via our access management system and outline your research questions. And once your application is approved, everyone on the project needs to complete a short set of courses, just a couple of hours in total, that cover both the data safety and compliance side, as well as some data and RAP-focused training as well. So briefly, the courses include the MRC's research, GDPR and confidentiality course, an introduction to the UK Biobank data, introduction to the RAP specifically, and also introduction of using version control and code repositories within the UK Biobank. Again, encouraging researchers to use code in a sort of controlled, reproducible, and safe way. So it is probably worth mentioning that the RAP is its own environment. It and it does come with a little bit of a learning curve when you start learning how to use the platform and when you first log onto it. But hopefully completing these courses will put you in a good place to then get started with your analysis. So in terms of first steps, once you're up and running on the RAP, the first thing that you usually need to do is dispense a project. The project is essentially your workspace within the RAP. All your data is kept there. Any files that you generate as a part of your analysis will be kept there as well. And it is a launchpad for any sort of tools or analysis you might want to use. This is briefly the screen that you're presented with when you're dispensing a project. Each project is associated with a unique UK Biobank application ID, but you can have multiple projects dispensed under the same application as well. You can control your billing as well, your billing accounts, and also you can control what data you dispense. So you can either dispense just tabular data, and that includes anything that can be stored in a tabular format. So the NMR metabolomics dataset, proteomics, GP records, questionnaires, things like that. Or if you are interested with some of the raw imaging, genomics, or accelerometry files, you can choose to dispense the bulk data as well, although that would make your dispenser slightly longer. So projects are really, really great for collaboration. As I mentioned, you can dispense multiple projects under the same application. So you can have a project just for yourself if you like to keep your analysis separate. But if you do like to share with your colleagues, you can share your project with anyone that is on the same UK Biobank application as you. You can control the level of access for users within the project, so you can give people just a view-only access or sort of more executable access where they can modify data and launch analysis as well. And you can do that either through the graphical interface as you get started, but if you do find yourself in charge of multiple projects, that can also be done computationally via the command line. One of the great things about the RAP is that there is a variety of tools available for every level of computational expertise. So you can immediately start investigating our data using sort of the more beginner tools, but as you scale up and level up, you'll be able to use the more complex tools, even perhaps build your own applications within the platform environment. So at the beginner level, we have tools such as the cohort browser and the table exporter. This is sort of what you're presented with when you want to use the cohort browser. Essentially, you select your data set of interest from your project, you click on the Start Analysis button, and then you have a visual interface that allows you to select your fields of interest, generate plots and graphs, and also generate table that contains individual level records for the participants you are interested in for each of the fields you have selected. And this can then be exported back into your project space for further analysis. The table exporter works at a similar level as well. You can launch it from the tools library within the app. And again, it can be launched in a very visual manner where you specify your fields of interest through a file format or a sort of a comma-separated format. You specify your desired output file format, and then you click Run. And then essentially it will extract the data you have requested and upload it to your project area for further analysis. Beyond the graphical interface, beyond the graphical interface tools, we also have interactive tools. So we have RStudio and Jupyter Notebooks, which allow you to code in R, Python, and Bash. There are multiple options when you request to launch an RStudio or Jupyter Notebook instance. One of the useful things is that you can load in snapshots. So, for example, if you have run through an analysis previously and have data loaded there or useful tools, you can essentially recreate that by using a snapshot, a safe snapshot file, which is a lot easier than alternatives such as using containers. We have multiple nodes, so single nodes that maybe you can use if you're working with data you have already, already exported. Spark is really good for really leveraging big data. If you're exporting lots and lots of fields and want your analysis to run quickly, the Spark cluster is probably the one to run. And of course, you're in control of the exact memory you want and the number of cores. So if your code is optimized for parallelization, for example, you can ask for a number of cores, and the cost is always there up front, so you'll know how much your analysis will cost ahead of time. There is also a command line interface that comes with a package loaded in that's specific to the RAP that has a lot of really useful functions. So the one listed here are just a small example, but it has a lot of useful functions for finding data and metadata, managing any jobs you're running, managing your project. All of that information is available on the DNA Nexus pages. But yeah, it is very, very helpful. And essentially, at the sort of more advanced end of the spectrum, if you have complex multi-step analytical workflows, the RAP does have really great infrastructure for supporting that. So you can build applets that you can then deploy from your project area. Applets can be developed locally in R, Bash, or Python, and the DX app workflow allows you to bundle up your analytical scripts with any dependencies you might have, such as packages, libraries, tools, and then you can build the applet and deploy it within the cloud environment. They can be deployed both through using the sort of the visual graphical interface that I showed you for the table exporter, or they can be deployed via the command line interface as well. So they might be quite a great solution. For example, if you are a computational scientist working in a lab with colleagues that are maybe not as computationally advanced as you, you can build this app workflow to allow them to interact with the data in a more graphical manner. So it's worth mentioning that you are, in terms of downloading data and files from the RAP, you are allowed to download summary level results and summary statistics. You are allowed to download graphs and images for publication with some caveats I'll mention in a moment. And you're also allowed to download any code or files that you have generated. You're not allowed to download individual level data, and that includes both tabular data or bulk data. If you do derive variables that are not present in the original data set, that are individual level variables, you can't export them out from the RAP, you can't download them, you need to return them to us, and then we can share them with other researchers. And also, when you are exporting things like graphs or code, be wary of including any sort of hard-coded individual data, so things like participant identifiers and things like that. Make sure that you are not exporting that out of the RAP. Obviously, everything that's individual level needs to be based to stay within the RAP environment. In terms of next stages and additional support, the Showcase website contains all the information on individual data fields. All fields have nodes detailing how the data was collected, as well as resources that can be very useful. And it's definitely very, very useful to consult Showcase ahead of starting your analysis. There is a lot of useful information there. We also have a community forum, which is a collaborative space for researchers to discuss how they analyze data on the RAP. And this is also where we post announcements and things like that. The UK BRAP documentation has a lot of useful advice as well, and documentation on applet building. There's a lot of webinars, both on sort of general access to the data, but also how to approach specific tasks on the RAP, such as how do I run a GWAS and things like that. And then finally, the UK Biobank data analyst team we have our own GitHub space, and on there we host a lot of notebooks and walkthrough guides on how to access data in the RAP. I have an example of that here. We have a number of notebooks, both in Python and R, that take you and guide you through things like accessing the phenotypic data, as well as slightly more specific applications, such as looking at the all-link dataset. We do have an NMR-focused notebook coming soon as well that we will upload to GitHub very, very soon. So please look out for that. We will probably communicate it through the usual channels. And that's it, really. I think myself and a couple of my colleagues are on hand to answer questions in the chat. And I'll hand back to Martin.
Yeah, Thanks so much, Laura, and thanks to Luke as well for giving us two really great presentations. Very, very helpful indeed. We're going to move on now to two case studies. So two speakers. Shelley Zhang is going to be the first up. She's a PhD candidate at Central South University in Hunan, and she's going to talk about research that's been done using these data focused on biological aging and age-related diseases. And then following on from Shelley, Mike Inouye is going to be talking to us about using these data for cardiovascular risk prediction. Mike is a professor of systems genomics and population health at the University of Cambridge and has done some fabulous work, as I mentioned. So Shelley will hand over to Mike just to save a little bit of time, and then hopefully we'll have a bit of time for a Q&A at the end and possibly bring in some of the speakers again. So I'll hand over now to Shelley. Thank you.
And thank you, Martin, for the introduction. And it's a really pleasure to be part of this webinar and to share our previous work on the metabolomic profile of biological aging. So over the next 10 minutes, I will walk you through our work published in Nature Communications in 2024. And in this study, we use large-scale UK Biobank NMR data to characterize the metabolomic profile of aging at the population scale. I hope this brief overview will give you a sense of how this data can be leveraged to study biological aging and disease risk, and perhaps spark ideas for your own research using the UK Biobank NMR data. The motivation and background of the study is that aging is the primary risk factor for most disease and functional decline. Accurately capturing biological aging is therefore critical for disease prevention, risk stratification, and understanding aging-related health outcomes. However, chronological age fails to reflect substantial heterogeneity in individual aging trajectories, motivating the development of biological aging measures. And recent multi-omics-based aging clocks have shown strong potential for quantifying biological aging and predicting disease risk. Among these, the plasma metabolome provides systemic and dynamic signals that closely reflect an individual's physiological state and overall health. And at the start of our project, UK Biobank has released the second version of the Nightingale NMR data, covering around 275,000 participants. With this large sample size and broad metabolic coverage and rich phenotype data, it offered a unique opportunity to study aging at scale. And this motivated us to ask a simple question: can a metabolomic-based model built at such a large sample size provide a more accurate and more biologically meaningful measure of biological aging? And our aims were twofold. First, to identify key aging-related metabolites and link them to disease risk and mortality. And second, to develop a metabolomic aging score that captures individual aging trajectories and explore its potential clinical relevance. With that background in mind, I'll briefly outline our analytical pipeline. And our framework has four key steps. First, we identify metabolomic biomarkers that best capture biological aging. And second, we examine their links to aging-related disease and assess potential causal relationships. Third, we integrate these signals into a single metabolomic aging score and evaluate its performance. And finally, we use the longitudinal data to characterize how metabolomic aging changes over time. And to be more specific, we started with 325 NMR biomarkers, many of which are highly correlated. Using all-cause mortality as a global aging endpoint, we applied Lasso-Cox regression in training and validation dataset to select a compact set of informative biomarkers. Next, we examined how these biomarkers relate to frailty-related phenotypes and aging-related disease. To move beyond associations, we integrate genetic data and applied Mendelian randomization and colocalization analysis to explore potential causal links. We then combined the selected biomarkers into a metabolomic aging score and compared its performance with established aging measures, including chronological age, telomere length, and the frailty index. And finally, using repeat metabolomic measurements from about 13,000 participants, we estimated a metabolomic aging rate to capture individual differences in the pace of biological aging. With that framework in place, now we'll move on to our main findings. And the first key finding is that we identified a compact set of metabolomic biomarkers that capture biological aging. Starting from the full panel of 325 NMR biomarkers, we selected 54 metabolites that together show the strongest prediction for all-cause mortality. And these biomarkers span multiple pathways, including amino acids, ketone bodies, and polyunsaturated fatty acids, inflammatory biomarkers, and a broad range of lipoprotein-related measures. Importantly, they were not only associated with mortality, but also show consistent relationships with other aging metrics, such as chronological age, the frailty index, and telomere length, as well as many frailty-related health outcomes. So taken together, this suggests that the biomarkers captured broad systemic metabolic changes related to aging, rather than signals driven by a single disease. To go a step further, we wanted to move beyond simple associations and ask whether these biomarkers might actually play a causal role in aging-related disease. To do this, we first ran genome-wide association studies for each biomarker and shared all of the GWAS summary statistics through the GWAS catalog. Because many of these biomarkers are highly correlated and the underlying genetic variants are often pleiotropic, we use multivariable Mendelian randomization, which allows us to model multiple related biomarkers at the same time. Using this framework, we identified 439 candidate causal links between metabolites and aging-related disease. Reassuringly, many of these recaptured well-known relationships, such as glucose with type 2 diabetes, creatinine with chronic kidney disease, and albumin with liver cirrhosis. At the same time, we also uncovered several more complex and pleiotropic roles of metabolites across different aging-related conditions. And based on these 54 biomarkers, we then developed a metabolomic aging score, which combines them into a single measure of biological aging. And here are two main takeaways. First, the score consistently outperformed other aging metrics, including a previously published metabolomic score, the frailty index, and telomere length in predicting all-cause mortality over follow-up periods from 1 to 15 years. And importantly, it even outperformed chronological age for short-term mortality risk prediction, particularly within the first 1 to 5 years. Second, the score could clearly distinguish individuals who develop aging-related disease at younger ages. People who were biologically older than their peers tend to develop disease earlier, and early onset cases showed higher metabolomic aging score at baseline across multiple disease. The score worked especially well for disease with strong metabolic components and further improved disease risk prediction when added on top of traditional clinical factors. Together, these results show that NMR-based metabolomics data capture meaningful and system-wide aging signals and provide a powerful way to quantifying biological aging and disease risk. So far, everything I have shown has been largely cross-sectional, but an equally important question is how the metabolome changes within the same individuals as they age. To address this, we use repeat NMR metabolomic data from about 13,000 participants with follow-up data and estimated an individual-level metabolomic aging rate, defined as the rate of change in the metabolomic aging score over time. We found that individuals who aged faster at the metabolomic level had a substantially higher risk of death. Specifically, those in the fastest aging group had more than a twofold
So have we lost Shelley? So I apologize for the technical problem there. I think we'll move on to Mike Inouye's presentation at this point, and then pick up with a question and answer session if we've got time at the end. So over to Mike, thanks.
Sure, no, thanks, Martin. Hopefully it's telling me I've got a lot of background noise. Hopefully that's okay. Great, well, thanks for the invitation to present to you today. I'm Mike Inouye, Professor of Systems Genomics and Population Health, and I'll show you a little bit about what we've done with both the genetics data and the new Nightingale metabolomics data in the UK Biobank. So our risk prediction interests are very much focused on cardiovascular disease, and just a little bit of background about cardiovascular diseases. So currently this is probably the, if not the top, amongst the top two sort of causes of death worldwide in both high and middle and low income countries. Frequently, what we see in cardiovascular diseases are atherosclerotic cardiovascular diseases, that is narrowing of arteries due to the accumulation of lipids and other things that create plaques in the arteries that may rupture, et cetera. And particularly people with coronary artery disease end up going on to have an infarct, that is a heart attack. So this is a major cause of death throughout the world. But cardiovascular disease aren't just that. They're broader. They include strokes, arrhythmias, heart failure, cerebrovascular disease, hypertensive disease, and aortic aneurysms as well. So the prediction of cardiovascular diseases is important because we can take preventative measures and hopefully, prevent events and deaths. So there's a long history of cardiovascular risk prediction right around the world. I'm giving just a key example here from Europe, but also there's UK specific, there's US specific, and many countries have their own schema. So within Europe, the European Society of Cardiology has a series of recommendations which depend on a particular risk prediction algorithm called SCORE2. And that algorithm is essentially predicting 10-year risk of a cardiovascular event in individuals who are otherwise not on preventative therapies, so particularly like lipid-lowering medications. So if you meet a certain threshold and you're in a certain demographic, the SCORE2 essentially gives you the 10-year risk prediction, and then you enter in, if you pass one of those thresholds, you have the option of entering into shared decision-making with your physician around whether you sort of are prescribed statins and lower your cholesterol levels, et cetera, and essentially reducing your relative risk by about 20% over 10 years. So it's not massive, but it's not insignificant either. And the UK has similar sort of guidelines essentially, but they depend on a slightly different risk model called QRisk3 or QRisk2 more recently. And the US has the pooled cohorts equation as well. So we asked the question, what is the incremental benefit of augmenting SCORE2 in particular with both genetics in the form of polygenic scores, which I'll assume people have some background in already, but if not, you can sort of Google it. Essentially, this is a genetic risk predictor. And then with biomarkers such as the Nightingale Health metabolomics, but also for completeness, some of the selected sort of clinical biomarkers that have been measured on the panel also available in UK Biobank. So we use the full sort of set of UK Biobank to do our modeling. We focus on individuals who are eligible for SCORE2, those 40 to 69 years. They do not have cardiovascular disease. They're not taking statins. And that brings you down to just under 300,000 individuals in the UK Biobank and just under 9,000 incident CVD events. And these are individuals who also have nearly complete risk factors as well, cholesterol, et cetera. So So we calculate score 2. We also look at individual and scores of biomarkers and polygenic scores as well. And then we take that forward into some modeling around the impact of screening, both population-wide and targeted, using population-wide demographics to recalibrate our risk models to a UK primary care population, so correcting for the innate, healthy participant bias in UK Biobank. So just jumping straight into the results. So just to clarify, these are pooled results. So I'm not showing you at the moment, but when you actually look at the scores, we do that in sort of half of the data. We develop those in cross-validation in half the data, and we use sort of the predicted scores from those cross-validations and merge that with a replication set completely withheld. We compare the performance metrics across both the discovery and the replication set, and essentially they're very, very similar. So we present to you here, for simplicity, a pooled analysis, but the full data, the full sort of results are in the publication. So here we're looking at the top 10 biomarkers individually. So for example, cystatin C, et cetera. And you can see that when you add these individual biomarkers to the score2 risk prediction algorithm, you do get incremental improvements in risk discrimination in the form of the delta C index, but that they're actually relatively modest. But there's actually quite a few of these, and you can see sort of the hazard ratios there as well. So the point is that here you see some signal even at the individual biomarker level for prediction of cardiovascular disease in this subset of UKB. But we're interested in scores here, so we also compute the scores, and here the Nightingale metabolomic scores are labeled as NMR scores because it's an NMR platform, and there's also the clinical chemistry, biochemistry markers as well. It's worth noting that the clinical biochemistry markers is a panel that's actually not commercially available, so really. You should be looking at the individual biomarkers for those, but we want to compare scores, so we've done that as well as a comparator. So essentially, what you see here is that when you add in the polygenic scores, the NMR scores, and the biomarker scores, biochemistry marker scores, you end up getting significantly larger delta C indices, still relatively modest, sort of 0.01 to about 0.02. But that as you progressively add more and more information, this all sort of is additive together, and you end up getting better and better risk discrimination as well. And you can take that forward using the score2 thresholds to actually ask, what's the improvement in the reclassification of risk. So do you actually increase the classification of risk for known future cases, and conversely decrease that for the non-cases? And here that's what's shown in sort of green and magenta on the right. And you see that you do get net improvements in reclassifications for both polygenic scores and metabolomics and for the biomarkers, but that it gets also progressively improved. So the question is, now that we've got this reclassification, we know our thresholds, what is the population-wide impact of that? So to do that, we essentially use a primary care population drawn from the CPRD primary care database in the UK. To essentially recalibrate our models to a primary care population, and that also reflects the demographics of the UK. So so here, if you I'll sort of skip through to the bottom here panel on in front of us. So as you go through and you ask, how many events, for example, or how many individuals, let's say, are designated as high risk. You can see that there in sort of the first column. Mainly what we're focused on is how many additional events will you prevent if you drop in a new biomarker into score2, and that's the sort of the second from the right, or sorry, the second sort of set of data points there in the green, yellowish green. So CVD high risk. Oh, sorry, middle, CVD prevented. So this is the green there in the middle. And you want to see that being pushed as far to the right as possible. So you can see with the PRSs, you get an increase, CVD events prevented per 100,000 people screened, of about, 50 or so. If you look at just the NMR scores individually, that gets pushed to about 90, 95. But that progressively, as you add these together, you end up getting more and more additional CVD events prevented over and above score 2. And the other main metric there is NNT, number needed to treat in order to prevent one event. That's at the far right in the magenta. And what you want to do there is essentially have that number as close to 1 as possible, right? Everyone you end up treating ends up being a prevented case, right? So you can see there that statins are not hugely effective, but at population wide they have an impact. But the NNTs are still sort of hovering around 22. But as you add in these biomarkers, you want to add them in such a way that that you don't push that number up. Ideally, you'd even reduce it. So you can see that largely, as you add these new biomarkers into score 2, you still sort of more or less hover around 22 and number needed individuals needed to treat in order to prevent one CVD event. Which is good. It means that there's a sort of, if the efficiency of prescribing medication is essentially maintained, but that you get this benefit of increased numbers of cardiovascular disease events prevented. If I may say, so the previous one was actually population-wide. So another strategy for deploying new biomarkers is to have a targeted strategy focusing in on intermediate risk. We did that here as well, and you end up seeing a very similar thing in terms of the CVD events prevented and the NNT. And it actually improves even over and above if you just take the similar number, like if you had people at intermediate risk and you just ended up treating a similar number of people just ranked by the score 2 algorithm itself. So we're doing significantly better than just going down by score 2 risk, which is a good sign as well. So overall, we were really quite happy with these results. Really quite surprised actually at some of the numbers that we found. And really the question is, at this point, this is still modeling, obviously. But the question is really, is it worth it for health systems to move this forward translationally, cost-benefit wise, technologically wise, right? So I think that's a question on everyone's mind at the moment. But anyways, we've published this work in the European Heart Journal there. Please do have a look, and we're of course very grateful to UK Biobank and Nightingale Health for enabling these really exciting analyses that hopefully will push the dial on cardiovascular risk prediction in the future. Thanks.
Really wonderful presentation. We've got just a couple of minutes, and I'd like to just revert to a very quick Q&A, but just perhaps after giving Shelley just a chance to summarize what she might have wanted to say at the end. I don't know whether, Shelley, you could come in just for literally a minute just to give us your summary of the work that you've done and where you see the field going forward in the future.
Yes, thank you. And sorry everyone, my connection was broken during my presentation. And I just want to conclude that our work shows how large-scale plasma metabolomics in the UK Biobank can be used to characterize biological aging at such a large population scale. And more broadly, metabolomics offers a powerful lens for disease research because it provides a systemic and relatively direct readout of physiological state and integrating the downstream effects of genetics, environment, and lifestyle. And with the NMR data expanding to up to 500,000 UK Biobank participants, and this resource will open the door to phenome-wide. We hope work could illustrate how exciting large-scale metabolomic data can be effectively leveraged to advance research on aging and complex disease. And thank you, everyone.
Yeah, thanks, Shirley. I'm so
also
pleased.
profiling improved disease. Yes, and thank you, Martin.
Okay, I'm so pleased that we managed to get your summary in at the end. So sorry about the connection. We've probably just got literally a minute. Luke, if I may just direct what time we have just to yourself and just bundle a couple of questions. Lora very kindly provided an overview of some of the resources that are available through UK Biobank about these data and how to analyse them. But you did mention the Kaggle, and could you just say a couple of words about that? There was another question about any QC steps required for further analysis of the data and how we interpret the data in terms of the units. So I don't know whether you could cover those 3 points briefly, and I think we'll probably bring it to a close after that.
Yes, briefly. I think that the UK Biobank will circulate after this. I think that Dan will circulate some information on all of these things, including that Kaggle worksheet. That is, when we finished the data set, we ran an in-person workshop for students and postdocs in London, in which we had basically a day-long course on how to analyze it, and that is the workbook from that. So that also includes some information on QC. In terms of QC, the data is there are some suggested QC processes that you can take out, some normalization things you can do in that workbook. Though in general, the QC is not. It doesn't require very heavy QC, and often that's more for sort of analysis reasons. There are outliers in the data. We don't think that those are poor samples. In fact, we think they mostly are actually biological outliers, but sometimes you might want to remove those because your data analysis might not be robust to outliers. And then finally, the units are all available on the showcase. So they are all in absolute unit quantification. But those details, actually, if you look up the individual variables on the UK Biobank showcase, or look at the showcase metadata files, that'll give the units in there in the standard UK Biobank unit field.
That's wonderful. Thanks, Luke. So I'm very grateful to everybody. Particular thanks to Luke, to Lora, to Shelley, and Mike for the excellent presentations. Thank you so much for putting that time in. For those, I'd like to thank Jenny and Dan at the UK Biobank for putting this session together. I'd like to thank the people who joined the webinar today for their contributions and the question time. I'm so sorry we've not had time really to address many of those questions, but I'm sure there'll be a lot of interest in these data going forward. And as we've seen from Mike and Shirley, there's some fabulous research that's possible with these data and massive opportunities for loads of people. So I do hope very much that this has been helpful to you and will stimulate some very exciting research going forward. So I'd like to bring this to a close and again thank everyone for their contributions today.